Hope everybody is having a wonderful Friday afternoon. Thank you for taking time to join us. I think I'm going to kick off this meeting. We have a long agenda to cover today. So with that, I think people is still join, but we will kick off the meeting. Thank you again for being here with us in this Friday afternoon. Welcome to the pre-application webinar for the funding opportunity announcement number PAR 24082. This is the 10th cycle of the Gabriella Miller Kids First Pediatric Research Program. We are calling for applications to submit samples for sequence. I am Marcia Fournier, a cancer researcher by education. I joined the program about 2 years ago as the program manager, and today I will be your presenter. We will start with a video as an example of the vibrant research community that Kids First is helping to build. So I will ask, we will start by watching this 3-minute video, and I will come back and then tell you our agenda for today.
The video URL is https://www.youtube.com/watch?G3G.
Before we get too deep into the video, just as a quick note, this meeting will be recorded. Please make special arrangements or take note if you would prefer not to be on video. Thank you.
I'd like to say thank you for the Kids First and include investigators for being here today. Data is driving discoveries that can impact lives, and the NIH is proud to collaborate with researchers internationally to make this come true.
I'm excited to be here at ASHG. Kids First is an amazing opportunity to sequence our patients and identify genes that might cause their disease, and potentially return those results to patients is an exciting way to engage research and learn more about why various genes cause congenital anomalies.
I am overseeing both programs, and I'm so glad to see we have a lot of intersection between those two programs. So we need to join the effort to conquer those rare disease cohorts, to create a power to identify some meaningful information from those data.
Really excited to be here. Very stimulating community. We all share the same goals, and finally we get to meet people that we have been reading the science about. Cannot be more happy. Thank you for inviting me.
The best part of this night is the fact that I've been able to learn from other presenters as well. And people have come to my poster to ask some questions, and they've made contributions to my work.
I'm really enjoying this poster session of the Kids First program and the Include program. We're having a lot of fun. We're learning a lot of really good science and making a lot of discoveries. So thank you very much for hosting us tonight.
What do you think about the Kids First program?
So Kids First, if it wasn't there, it would have to be invented. So I think it's really important because pediatrics always comes last, and it's really important to have like a program where kind of kids really come first, and we can ask questions that we typically don't have space for.
And I'm so excited to be here tonight and discuss all the exciting findings and great research that we all have been doing, and how useful GMKF cohorts have been, and the need to do more sequencing, more better phenotyping, and discuss what all we can do as a community moving ahead from here.
It always lifts my spirits watching this video. And this is the type of vibrant community we are building at Kids First. And today, you're going to hear about the funding opportunity announcement for sequencing cohorts. So we can come back to the slides now. Thank you. We're going to go to the agenda today. Next slide. So the agenda today will be, I will talk a little bit about the program, the application guidelines, and also I will cover a little bit of the Kids First resources. I want to ask you guys to keep your cameras off for now, but we will ask you to open your cameras if you want. If you have questions, ask questions throughout the event in the chat box, and we will answer all questions at the end of the event, but you may be able to ask questions anytime using the chat box. And then in the end of the event, during the Q&A, I will ask you to open your cameras if you want to. Next slide. So how it started, the Kids First program started with Gabriella Miller advocacy in pediatric cancer. And part of her legacy is in 2014, the Gabriella Miller Kids First Research Act, which authorizes $12.6 million a year for 10 years to the NIH to support pediatric research. And part of the vision of the program is fostering collaborative research and supporting data sharing. Next slide. Why structural birth defects in childhood cancer? The association across pediatric conditions is the scientific hypothesis for this program, especially with multiple reports on shared mutations and drug targets across pediatric conditions like BRAF, MAPK, and ALK, as well as recent reports on the association between birth defects and higher risk for cancer in child and adults. So it makes sense to create a resource where researchers can study across pediatric conditions, including structural birth defects and childhood cancer. Next slide. Kids First current priorities. I want to tell you a little bit what we prioritize. And so one, we investing on generating data from childhood cancer and structural birth defects. We want to add multiple data types, such as genomics, epinomics, proteomics. We want to add longitudinal data, phenotypes, clinical data, and outcomes. We want to add diversity of disease and diversity of participants in cohorts. And we prioritize allowing broad data sharing with individual level genomic data available through dbGaP through controlled access requests. Two, second priority, we are developing a data resource. We want the data be findable, accessible, interoperable, and reusable. The data resources are enabling cloud computing for data discovery, data access, and data analysis. And we are developing new analysis tools and workflows that will enable the research community overall to find the data and use the data. We are promoting cross-disease collaborations and establishment of interoperability among cloud platforms. The third priority for the program is to support expert-driven activities to increase the value of the Kids First data. So we want to engage the research community in activities such as data integration, data curation, data harmonization, development of analysis tools and workflows that not only are useful to your research, but also useful for a broader research community to use as part of our resources. So now, next slide, I want to talk to you a little bit more. Let's get into the EXO-1 mechanism. So the Kids First data generation cycle is empowered by the EXO-1 mechanism, along with the genome sequencing centers, the Broad Institute and Hudson Alpha. And thank you, by the way, for representatives of both sequencing centers be here today. And the Data Resource Center also, thank you for the Kids First Data Resource Center members that are here today. But really all starts with yours, researchers, your patient cohorts. So you are here in the proposal submission, submitting to the NIH EXO-1 mechanism. As any EXO-1 grants, this will go through a pre-review process to establish scientific merit. And after the review process is completed, projects are selected by the program. And I want to start then mentioning two key components of how we prioritize projects for sequencing. One, we prioritize cohorts that allow broad data sharing. And two, we support cohorts that have annotation of phenotypes and clinical data. So far, we supported 70 EXO-1 cohorts through the years. And once the projects are selected, the cohorts go through samples go to sequencing centers. And then after the data is generated, the Kids First Data Resource Center will harmonize the data and deliver the data back to the EXO-1 investigators. EXO-1 investigators will have 6 months exclusivity with the data. And then after that 6 months, the data is made available to the public through dbGaP and the Kids First portal. And this is the biggest milestone for us in the program, is when we get the data in the public domain through the dbGaP and Kids First portal, and it becomes part of our resource that will empower the research community. And 33 studies are already made available through the portal and dbGaP. Now, next slide, I would like to talk a little bit about the timeline. After this webinar, you will have an opportunity to send us a letter of intent. It's not mandatory, but it's welcome, because then for planning purposes.
The letter of intent deadline is February 13th.
But even if you don't have time to put that letter of intent in, you can still apply. The deadline is March 13th. And then after you apply, I want to give you a sense of the timeline for when you're going to hear back from us. You're going to hear back from us multiple times, probably asking for questions and information, but really the decision and notifying recipients and the EXO-1 orientation for the selected projects is expected to happen early September, October, so fall 2024. So if you're applying on March 13, you should expect the decisions late summer, and then the notifications to the recipients early fall. Next slide. I would like to highlight key points for the PAR 24082. Data sharing is critical for Kids First, as you already heard multiple times. But as part of the application, there is no requirement for having a data sharing plan separately. Your data sharing plan will be part of your research plan. So the research plan is a requirement where you're going to tell us representation, diversity, inclusion. You're going to describe your cohort, the family tree, phenotypes, clinical information. And then you're going to describe your data sharing plan. Other attachments that are also a requirement include the institutional certification, and I will go over in detail what you need to do, sample information, clinical and phenotypic data, and even though family structure is optional, I highly recommend for cohorts that involve family members. All right, so next I would like to tell you about the sequencing centers. The goal for the sequencing centers, and thank you again, Broad and Hudson Alpha for being here today, is to generate high quality whole genome sequence and variant data from childhood cancer and structural birth defects. But what are these expectations from the proposals? One, the DNA samples must be sufficient quality and concentration for whole genome sequence or long read, specifically PacBio Review, which is the main technology we are using now. And RNA samples, in case of you're requesting transcriptome for tumors or affected tissue, must be sufficient quality and concentration for RNA-seq. And samples will be ready to ship by the end of this calendar year. So I want to emphasize this third bullet, especially this year. As you heard, this is a 10 years program, and this is the 10th cycle. For this year specifically, is a must to have your samples shipped by the end of the calendar year. And this will be a key point for prioritizing and selecting project this year, is that in your proposal, you show evidence that you can ship the samples by the end of the calendar year. Study design. Next, our goal is that you tell us your study design, your sample size, your family structures, and if this is sufficient to lead to a genetic discovery. So the study design will tell us how you're going to use this data to achieve a scientific progress or discovery. So the expectations, we prioritize large sample sizes, so we encourage researchers to collaborate with other investigators to pool samples together and propose large cohorts. And for non-trio designs, describe the probands and if any affected or unaffected family members are proposed for sequencing. Again, we will encourage you to describe it as much as possible. But the main thing is how you're going to use this information to solve a scientific question. Your scientific question is important, and this project, even though it's a sequencing project, it is driven by a scientific hypothesis and a scientific design of your study. Next slide. So regarding the, you seeing a screenshot, and this is a table you must submit with your proposal, describing information, describing your sample. So the DNA, what is the sample origin, blood, saliva, tissue, number of samples, extraction method, concentration, etc. So that is how you're gonna not only written in your proposal in the research plan, but also submitting this table with specifics about the sample information. And that is a mandatory must to have part of your proposal. Next slide. Analysis plans. So the goal is, again, to have investigators to demonstrate it that the proposed project has an adequate research design for a genetic discovery, and that EXO-1 applicants are prepared to perform this analysis. So even though the EXO-1 is focused on generating data, the analysis behind the data is important for the project evaluation. We want to know how you're going to use the data and have a feasible analysis plan. So the expectation is that not only you can demonstrate that you are solving your question, your scientific questions, in addition that this data will be enrich our data resource, but it's still your proposal will be driven by your scientific question. So consider partnering with other research teams that has expertise in data analysis to develop your analysis plan. And researchers are encouraged to use cloud-based infrastructure, include Cavatica, for their data analysis and the analysis plan. So next, I will talk to you about the data sharing. So the goal is to make data generated by Kids First as accessible and usable as possible to the research community. Again, there are two main portions here. One, demonstrating this data will answer your scientific question. Two, demonstrating this data is valuable for the research community, because as you heard, the big milestone of this program is when your data is part now of that portal and part of a broad data resource that will empower pediatric research community. So the expectations is that the data allows for individual level sequence data sharing, and you provide relevant phenotypes and clinical data, because the genomic data goes so far. And really for empower analysis is key to have well-annotated phenotypic and clinical data. So samples, again, that are consented in a way that allows broad data access and data usage, prioritize it in the selection process. So next slide, I will talk now about institutional certification, which is required for your application. And you can download the current NIH institutional certification template. And I think we will make this link available to you at the chat box.
The link URL is https://osp.od.nih.gov/scientific-sharing/institutional-certifications/
So download, and but I will go actually now page by page to tell you key information you need to know about how to fill up your institutional certification. So next slide. Institutional certification, page 1. When, like you see on the top of the page, name of GPA as Valerie Cotton at the NICHD. And then a second thing I would like to highlight on the page 1 of the institutional certification, as you see here, the screenshot under the institutional that are contributing samples, list all sites contributing samples on that area of the page 1 of your institutional certification. Page 2 next. So on the next page, there are 2 areas I want you to pay attention. One, when you are asked, the individual level data are to be available through check 1 that is on top of the page. 2, you mark controlled access, because we want to protect privacy, and the individual level genomic data I expect it to be controlled access. Down below the page, when you have to check for the genomic summary data, you leave controlled access unchecked, because the keeping unchecked the unrestricted access to genomic summary results is what it is expected for us. Page 3. So again, the screenshot of page 3, data sets that are consented for general research use, or GRU, or health medical biomedical, HMB, will be prioritized over data sets restricted to disease-specific use, as highlighted on top of the page. And on the bottom of the page, select general research use with no modifiers is expected for individual level data sharing, unless specific users are clearly prohibited. So next. So if your institution cannot have the final institutional certification done by the deadline, you can still apply your proposal using a provisional certification. But either a provisional certification or a full certification are required at the time of the proposal is submitted on March 13. And your full certification will be required for the selection of projects and prioritization process of the projects. So if you submit a provisional certification, we will reach out to you requesting a full certification before the selection process is completed.
For a list of standard DULs and modifiers, please review the institutional certification template at https://osp.od.nih.gov/scientific-sharing/institutional-certifications or https://wp.od.nih.gov/wp-content/uploads/standard_data_use_limitations.pdf.
Okay, any questions about that, you feel free to start putting in the chat box. We will try to answer as many questions as possible in the end of the event, but you can already put in your questions if you have questions. So let me give you now some examples of data use limitations and modifications that will limit broad data sharing. Therefore, we would not prioritize projects with such type of limitations, such as disease-specific consent group, which restrict access to a specific disease area. Or, for instance, a RRB modifier, which will require the requester to provide documentation of their local RRB for approval of the proposal researcher. Usually, the RRB modifier box is checked as an error. So, check that with your institutions to make sure, because these RRB modifiers usually are not prioritized. And COL modifiers, which require collaboration with the original investigator, is also not prioritized over. So this is data limitations and modifiers that would limit broad data sharing and are usually not prioritized during the selection process over projects that allow broad data sharing.
For a list of standard DULs and modifiers, please review the institutional certification template at https://osp.od.nih.gov/scientific-sharing/institutional-certifications or https://wp.od.nih.gov/wp-content/uploads/standard_data_use_limitations.pdf.
Just as a reminder, every requester of the data and the institution must agree to the terms of the data use certification, which verifies that the requesting PI is accredited within the institution. The institution is aware of the project for which the PI is proposing to use the data, and that the institution has all appropriated security measures in place to manage and maintain controlled access data sets being retrieved.
To access data use certifications, visit https://osp.od.nih.gov/wp-content/uploads/model_duc.pdf.
So let's go over now the clinical and phenotypic data. As I said, well-annotated data empower analysis and is a priority and commitment of the Kids First program to prioritize projects with reaching information about the phenotypes and clinical data. So the expectations is that these basic data elements are provided at the time of proposal. The next slide is a screenshot of a table that I have to be submitted with your proposal with the information about demographics, clinical information, other phenotypic information, and family medical history. And as mentioned, proposals with information on phenotypes and clinical data will be prioritized over projects with limited information. Next slide is a screenshot of biospecimen and phenotypic data elements as an example of elements that the basic elements that we require submission for projects. So projects must provide the minimal descriptors of cohorts, but we highly encourage you to customize the data elements to your cohort. That is highly encouraged because that will be the most useful when we are harmonizing the data for data sharing. And next slide, pedigree table is not required, but encouraged if your cohort involve families. Tell us how many families per, how many samples per family, and how many of the family members are affected or unaffected. So now I would like to go over examples of research projects that have been supported in the past. And I encourage you to go to the Common Fund website and the Kids First page to take a look on the EXO-1 projects that have been supported in the past, but examples are projects that are proposing discovery of biology, genetic variants. They're proposing whole genome sequencing or long-term sequencing approaches, integrative omics approaches, including transcriptome, epitomics, and methylation, chromatin accessibility, proteomic data. Projects that are increasing diversity within our data sets, but also representing conditions not previously sequenced by Kids First. But also, if you are increasing the number of samples in specific conditions we already have data, is also welcome. And many alternative approaches may be proposed. One reminder is once the projects are selected and during the orientation process, we tend to finalize the project design in collaboration with the sequencing centers, the Data Resource Center, and the NIH staff. So the take-home messages are read and follow guidance.
You can find selected EXO-1 projects at https://commonfund.nih.gov/kidsfirst/X01 projects.
And the next slide, please. Read and follow guidance from the program announcement. EXO-1 gives access to generate genomic data performed at the Kids First sequencing centers, which are the Broad Institute and Hudson Alpha. And the Kids First EXO-1 does not fund the data analysis, but projects showing a strong data analysis plan are prioritized. So the program prioritized proposals with inclusive, diverse cohorts, bringing rich phenotypic and clinical information and allowing broad data sharing. And last, I want to mention to you as that the selected projects to date are not listed in the NIH reporter. If you want to take a look at the EXO-1's information that was supported in the past, again, go to the Common Fund website, the Kids First data, and we will post this link at the chat box.
The link is https://commonfund.nih.gov/kidsfirst/funded-research.
Okay, so I'm going to give a pause here and see how we are on time. And I want to make sure that we have time to go through the over everything. So I will spend probably another 5 or so minutes talking a little bit about our data resources. And if you have questions about the application process, feel free to put in the chat box, and we will answer as many questions as possible in the end. So as if you remember in the beginning, the big milestone for the program is when the data is now available through dbGaP and the Kids First portal. So all Kids First projects are registered, authenticated, and approved through dbGaP. And all dbGaP data access requests will be processed by the Kids First Data Access Committee. Why is this important? Because having a actually dedicated committee to approve individual level data for requesters speed up the process. And usually requesters have data approved in 1 or 2 weeks. So the Kids First Data Resource Center is the NIH designated repository for the Kids First data. Next slide. So Kids First as a priority is developing a cloud-based resources that it will enable pediatric researchers through our cloud platforms, the Kids First Data Resource Portal, where researchers can explore, discover, and connect with data.
Visit the portal at https://portal.kidsfirstdrc.org.
And the cloud platform Cavatica for computing large-scale workflows on genomic data files, analyze and visualize data, and share data. So these tools through as an entry point to data resource portal where you can query, search, discovery data, build data sets, combine data sets, and other tools that we are making available in frameworks that will enable even experienced bioinformatics people. Our goal is to enable the community to use the data and analyze and visualize the data to foster your research. And that would not be possible. It takes a village. I want to acknowledge all the investigators and institutions participating, developing these cloud-based resources. Next slide. What is available today? And actually, this is available to you. Even if you don't have a EXO-1 project, you already can be part of the Kids First community by accessing the data as a secondary user. So as I mentioned, 33 new pediatric cohorts are released to date, including over 27,000 genomes available from affected children and families. And the body map here shows the different body parts where the data is already made available and the conditions that are included in the Kids First data resource. We already had approved 620 plus requests for secondary use of the data. and the data is available through dbGaP and a request for controlled access. And as I said in the beginning, it takes 1 or 2 weeks to get that access. So next slide, I would like to give an example of a recent released data set, which is across multiple pediatric conditions. And at the Kids First, we encourage this type of projects because we believe the cross-pollination across pediatric conditions to empower discoveries. And this was a result of a collaboration between multiple institutions, 6 different institutions collaborating to put this data set of over 200, 400 participants, along with phenotypes and clinical information. So we encourage this type of collaborative efforts when we prioritize projects to support. Next slide. So the data at the portal, you have access already to 22 birth defects studies and 11 cancer studies. And again, this is available to a large research community. The portal has reported over 3,000 users and across 50 unique countries represented in the portal users. So it's not only available, this data is not only available to be used in the United States, but internationally. And next slide, I would like to highlight the genetic variant browser, which it's available through a simple registration to the portal. So I want to stop here and just go over a little bit the tiers for data access. One, you can have access to summary level data by just, if you have an email, is a quick registration. Actually, I have a story. One of our program consultants came to me in a conference, frustrated that we needed registration for access of the data. And I challenge our program consultant to go through the registration process, and thank you for our team allowing us to challenge each other, and he accepted the challenge. And for his surprise, in 15 minutes, not only he had registered, but he had access to the summary level data, which is available through the portal and is an entry point. So you can interact with which conditions, which data we have at the summary level. And the variant browser, you can query variant across studies, narrow down variants of interest, identify number of participants with a given variant. And if you have a gene of preference of interest, you can filter variants based on a gene name. So this is all available through a simple registration that only confirms you're a real person and you have a real email. It doesn't even need to be institutional email to have a summary level access. Okay, so I told you I want to leave at least 15 minutes for questions. So the next slide. You may be asking, Marcia, but I don't have funds for cloud computing. Oh, well, NIH has an amazing program through the Kids First program sponsoring cloud credits. So it's a simple application through an email. For more information, go to our GitHub page, and we will make this link available to you at the chat box.
The link is github.com/kids-first/cloud-credits.
We already have 18 projects approved for cloud credits, 10 for structural birth defects, and 8 childhood cancer projects. And there is an opportunity to follow up for more credits. So this is great. If you have or not experience with the cloud computing, either way is a great opportunity to have funded, sponsored by NIH in form of the cloud credits through the Kids First Cloud Credit Pilot.
Email questions to kidsfirst@od.nih.gov.
So next slide. So I want to mention this funding opportunity because, as I told you, the EXO-1 mechanism is focused as a laser on data generation. And you may be asking how I'm going to analyze the data after I have the data. So this is a RO3 mechanism that the Kids First made available, PAR 23075. So the Common Fund has established this mechanism and for specifically focus on Kids First data analysis. But even if you don't have a EXO-1 project and you would like to do secondary analysis of Kids First data sets, this is also a mechanism, a relevant mechanism. If you would like to develop tools and workflows that you believe not only will be useful for answering your scientific question, but also would be useful for a broader research community, this is also a relevant funding mechanisms for you. So I welcome you guys to check it out. There are several entry dates for this mechanism, so check it out, PAR 23075. Last but not least, I would like to tell you about the short courses. This is a mechanism that for research education. And it supports training, short courses, and also training that would enhance diversity inclusion for data users. So application deadline is February 14, and I welcome you to check this opportunity.
The opportunity number is RFA-RM-23-014.
Next slide. Just a reminder, the deadline for the EXO-1, the Kids First EXO-1 fiscal year 2024 is March 13, and we will make the link to the PAR 24082 available in the chat box.
The link is grants/guide/PA-files/PAR-24-082.html.
Next slide, I want to leave you with some contact information. If you have questions about your institutional certification and any other questions before you submit your application, feel free to reach out and we will be happy to support you.
You can reach out to Marcia Fournier, Program Manager, at kidsfirst@od.nih.gov. Valerie Cotton, Genomic Program Administrator, Deputy Director of Data Science and Sharing at NICHD. Jamie M. Goodry Oville, Director at NCI Office of Data Sharing at nciofficeofdatasharing@mail.nih.gov. Email General NIH Genomic Data Sharing at gds@mail.nih.gov.
Answering your questions, and with that, we'll welcome your questions. So we will stop sharing, but thank you again for being here today. Thank you for your questions, and we will now answer as many questions as we can.
After the webinar, additional questions can be emailed to kidsfirst@od.nih.gov.
So let me take a look here on the questions. Wow, we have lots of questions. So, okay. So you mentioned singletons, meaning that now the program is more open to non-trio studies. in the past, single-dose studies did not fare well with this mechanism. So what I have to tell about that is that we are still prioritizing trials, duals, and families. But depending, again, on your scientific question and the goal of the scientific question, the goal of your project, if singletons is what makes sense and it makes a strong scientific justification behind it, it is welcome as well. So we are not opposed to proband-based studies as far as it comes with a strong scientific justification. Second question here, can research projects from institutions outside the US apply? Yes, the answer yes. So this is a mechanism that allows international applicants. Make sure to look at the description, the program description, because there are some steps. Your institution will have to be registered, and it takes few weeks to register the institution. But as far as you follow the steps described in the funding opportunity announcement, the short answer is yes, international institutions and international PIs, principal investigators, can apply to the Kids First Exo-1 mechanism. Next question.
Marcia, can I just add one thing to that as well, actually, as a sequence?
Absolutely.
Yeah, hi, everybody. This is Namrata Gupta from the Broad. Happy to work with some of you already. Just wanted to clarify another point is that we also receive a lot of samples out of the US for sequencing. There's, as Marcia said, we would require like a few more documentation, like some sort of, the attestations for GDPR, and we have templates and stuff that we can send off to you. So we can definitely do it. If we know in advance, the sooner we know, the better, because as Marcia said, it does take more time to get all the ducks in a row before those samples can come in. So just to keep that in mind, that if you do have that up front, which I imagine you will know as you're applying, for this XO1 mechanism. So, but if you have any other questions, feel free to reach out to us separately, and we can even provide you the templates up front so you know what you're looking at.
Thank you, Namrata. And actually, I would like to welcome everybody to open your cameras now as we are here in the room so I can see your faces. But this is a great question. And actually, I want to add to what Namrata said, that if you are an international institution or international principal investigator, also in your proposal, make sure you make the case that you have experience shipping internationally your samples, because this will be part of how we'll prioritize. Remember, is that making the case you can ship by the end of the calendar year. So having some evidence that you've done in the past that your samples can reach the United States is kind of like important case to make in your proposal in case you're an international institution. Any other questions? Okay, so let me go back to my question list here. So I'm not looking at you guys now, I'm looking at the questions here. Can research project, okay, so that was already gone. Next question is regarding general data questions. What kind of data deliverable are available during the 6 months embargo period? Okay, so you wanted to know, so basically, the sequence data and the harmonized data is delivered back to you by the Data Resource Center. So after the data is generated, the Data Resource Center will be responsible to compile the genomes, phenotypes, and clinical data. We actually go through a process that we call clinical data review, which really requires collaboration with the laboratories, the NIH staff, and the Data Resource Center personnel. We collaborate to compile this data and make sure the data is accurate. We want to put data out there, deliver back to you a good, excellent quality data. So that's why we go through this clinical data review process. And then you get this data delivered to your door, the sequence, the raw data, I believe, And if anyone from the DRC wants to jump in and say, add to what I'm saying. And also you get a few weeks later, the harmonized data. But even before the harmonization is done, we want to get this data in your hands as soon as possible, because we know that a lot of investigators already have a plan and they want to start working with the data even before the harmonization is completed. So that is what you get. And then the minute you get the data delivered to you, that's when the clock starts, 6 months. And then after that 6 months embargo period, the data then will be processed to be published in the portal. And if you have more questions about that, feel free to open your mics now. If anyone would like to make any comment about that or other questions. Okay, so let me go then to the next question. Can we request 1 nanopore proteome flow cell per sample with access to raw signal data for methylation analysis? And can we request Illumina whole genome sequencing parallel for mutation detection and improved halotype construction? Where the data deposit, our current protocol is designed for dbGaP deposition. Yeah, so let's go per. So in general, I will ask the sequence centers to answer the more specific questions. But as I mentioned, we welcome projects that are integrating multi-omics, whole genome sequence, methylation profiles, as far as has a strong research justification. Even though this is a data generation program, we want to make sure that it has a scientific rationale behind anything you're asking. So that is the far most important thing. But then comes the technology. Obviously, we are limited to support the technology that are available through our designated sequencing centers. So I will, the specific question about the proteome ION flow cell and the Illumina whole genome sequence, I'll let the sequencing centers to chime in on that.
Sure, happy to on the Broad side. The one that's being requested, the ONT, we actually at the Broad have the Revios from PacBio in production. So that's the one that we're doing for the current GMKF year, as well as for AOU. That's the one we're using. We are looking at ONT and kind of, starting to see what that looks like, but it's still kind of in development and we haven't sort of, put it into kind of a production setting. Now, in terms of, requesting the amount of data, whether it's ONT or PacBio, I think that's really up to the program to decide, in terms of what the funds are, what the scientific aims are. So we as a sequence center are, more than willing to, sequence whatever the program allows at that level. So like, for example, currently our PacBio long read offerings are sort of at the 12x or 24x coverage modeled at different pricing. So, we can work with you and the program to decide like what's the best for you at that level. And then same thing, I guess the second question is also really for program to decide. So we're more than happy to run, a whole genome in parallel with a long read. So that's, there's definitely people who do that for different reasons. So I think that's, if there's anything else specific from the Broad, but that's kind of where we stand regarding that.
Yeah, and on Hudson Alpha side, we're the same. We offer PacBio Revio, but we are not currently offering Oxford Nanopore.
And as just emphasize again, the most important thing is the research rationale and why you need it to answer your scientific question, the different omics that you're requesting. And there is a fair amount of collaboration once the projects are selected to refine the projects and collaborate with the sequencing centers, the data resource center, and NIH to refine the study designs and the technologies choices. And then I think last question I have here, will proposals for long read transcriptome sequence be considered? Hudson Alpha has performed this type. Okay, so this is, I will also, again, will be part of refining projects, and we are able to support as far as is supported, the technology is supported by our sequencing centers. While we welcome, I think the mainstream for long read now is the DNA long read projects. And, but again, is depend on your scientific rationale behind requesting that and the technology being available.
Hudson Alpha is currently supporting long read RNA on the review.
All right, so I think we're getting to the end. Okay, is spatial transcriptome available in either sequencing center?
I think, Marcia, we're definitely working on it. I think there's a lot of work and development right now in that space. I would be surprised if it would be ready for this year's mechanism, though. So just to be very transparent about that.
And Hudson Alpha does not have that option.
All right, we are coming to the end of our session. I think we have time for maybe one more question before we close. I think there are a few other questions here. Let me see. So where exactly are sequencing centers located in the US? And the size, okay, also, so you want to say where your locations, Namrata and Jane?
Oh, yeah, we're in Cambridge, Massachusetts.
And we're in Huntsville, Alabama.
Okay, and also there is another question here about size of cohorts. Great question. So there is no size. I won't tell you a number, yes or no. There is no size specific, but we encourage large cohorts, and by large cohorts is hundreds, thousands of samples. So we encourage collaboration among different sites to bring large cohorts on your proposals. But again, the top far most important thing is that makes sense for your scientific question. Let's say if you have a rare condition that only has 12 individuals around the world, like it would not make sense that you propose 1,000 samples, right? Because you don't have it. So everything needs to make sense for your scientific proposal, scientific goal. So that's why there is no number, but as far as is allowed, and through collaborations, you can achieve large cohorts. We welcome that. But we also respect that the scientific rationale is the most important for us, that it needs to make sense. And so if you're bringing a new condition to the data, and if it is super rare, so these all needs to make sense. All right, so thank you so much for being here again in a Friday afternoon, taking time to be with us, and I'm going to be excited to see your proposals coming in. If you have questions before your submission, feel free to reach out to me, and also we'll love to see your intent letters for planning purposes. So with that, I will close the session today, and thank you all. Have a good weekend.
Thank you.